Okay, so in this part, we're going to look at antibody-mediated immunity, part 3. And as we saw before in part 2, we had a number of factors that and proteins and receptor interactions between the cells that we saw as very important. And what I want to point out is that there is a certain organization of that, of those molecules. The MHC2 T cell receptors and all. We've seen that as sort of like the schematic before. Now I want to just point out is that in recent years, in the last several years, we've really understood what is actually the nature of that interaction between the cells themselves, the antigen processing cells, and the T cells. And what I want to show you in this sort of schematic here, you can see that in fact when two cells interact, they don't interact with the entire membrane. They interact with just a portion of the membrane, as you can see here. Let me just get this pen here, color pen here. You can see here they interact with a portion of the membrane. And what has been found when you fluorescently label those molecules like ICAM and the MHC2, for example, what you find is that there's actually what's referred to as it's called a synapse, or an immune synapse. Much as you think of the interaction between two neurons, it's called the neuron synapse. This has been given the name of immune synapse between the two cells. And what you see, if we were to take a cross section of that, we'd see that in the middle of that synapse we'd have a region that's heavily concentrated with the MHC2 and T cell receptor interactions. And then around it are the adhesion molecules that surround it here, cross section. And what this gives you is this bullseye, in which, like in the yellow here, this yellow region here, you can see here, and B is where the T, if we were looking, in this case, if we were looking edge on into the B cell, we would see the MHC2s, and then we'd see ICAM. If we looked on the T cell receptor side, we would see T cell receptors in the middle and LFA1 on the side, and it forms this kind of bullseye. And you can see down here, if we look at this is sort of a dimensions of it. You can see that on the T cell receptor, you have in the middle there, it's called the supramolecular attack complex, or this. We can see in the center of here, in the center of that, you see all of these interactions here: the T cell receptor, the MHC, CD28, the B7. All of those are in that middle part here. And then on the outer part, what you find is you'll find these adhesion molecules which surround it, forming this bullseye. And this allows for tight contact between the cells, and it means that there's many interactions. There's not just one MHC2 interacting with T cell receptor, but many MHC2s that are sort of corralled and interacting with many T cell receptors. And this allows for direct interaction of those peptides and increases the ability of those MHC2 peptides to find the T cell receptors and interact with them. Because the interaction between them individually is not very strong, it's maybe on the high nanomolar range, and so they would be fairly dynamically coming on and off. The Kate dissociation constants would not be strong enough to maintain a really high affinity interaction. But if you have many of them in that center, you can form a number of different contacts, like several different hands, that would strengthen that complex and make that interaction such that it would now have a certain amount of those T cell receptors and MHC2s that would allow for the signal transduction signal transduction pathway to be actually propagated through the cell. We look at another. This is another picture of this, and we can see here in the inner part, the green. So this is of a T cell receptor. This is a T cell. So if we sort of cut away the B cell interaction. So this is a T cell interacting what, if we were the B cell, this is what we would see. And what we see is in this green is the T cell receptors, CD4 and CD28 are all in this green part, whereas on the red part you find the interactions are the LFA1 interactions are all in here. So corralling it and sort of increasing the concentration of that interaction between the two cells and the potency of that interaction between the two cells. And so what is the nature of that? So for the next few minutes, for a little bit, what we're going to look at is what's the nature of that interaction that gets transmitted into the cell. Why is that so important? And we've shown that you get certain transcription factor, or certain cytokines are produced by the cells when they receive these signals. And so what we want to take a look now is some of the specific signal transduction pathways that are used. It's really important to understand this, as we're going to see later, because there's a number of drugs that are involved in interrupting these pathways in order to suppress the immune response. And so if you understand these signal transduction pathways, or the basic concepts of them, then you can understand how these drugs could work, and when they might be useful and when they're not useful. So there's a bit of an alphabet soup in all of these signal transduction pathways, as you've probably been aware by your reading and by in classes, that basically there's a shorthand, and it can be kind of mind-numbing in terms of trying to figure out what's what, keep it intact. So what I've done here is I've listed some of the key molecules. This is the shorthand, the acronym, shorthand like here, followed by the full name. So one of those is phospholipase C, or PLC. Another molecule is diacylglycerol, or DAG, protein kinase C, PKC. And then there's 3 transcription factors we're going to take a look at. Nuclear factor activating T cells, which is involved in the regulation of IL-2 expression. Another molecule, activator protein 1, which is a partner transcription factor that works with NFAT, and is also involved in IL-2 production, as well as several others. And then another transcription factor, NF-kappa B, nuclear factor kappa B, and this is in the regulation of not only IL-2 but other transcription factors as well. There's another molecule we'll see as an inhibitor that inhibits the action of this molecule, NF-kappa B. So the I is inhibitor of NF-kappa B. So these are sort of the molecules you'll need to know, and so you want to maybe keep that in mind, and as you're looking at the pathway, refer to this to keep yourself straight. So we've seen this before. Once we start I've listed here the interaction. You can see here the interaction between the MHC2 in the peptide and the T cell receptor. And the peptide is in the middle, which makes the interaction. And then CD4 comes along and is basically sort of holds this complex together. Now, I've up until now just had CD3 as GN1 molecule, but actually CD3 is composed of several different subunits. Epsilon, delta, gamma, and zeta. And particularly the zeta one is very important because it's involved in the signal transduction pathway. And in fact, this complex, this T cell receptor, this is referred to in total as the sort of total T cell receptor, is very complex. It's one of the most complex we know of. But this is what happens. We're going to go through those pathways that are involved when these are activated. So when this happens, when you get this complex formed, a molecule LCK, which is a kinase, I think it's leukocyte, sorry I didn't mention that on the other one, leukocyte kinase, will bind to CD4, and what it does is it phosphorylates the zeta chain of CD3, and that recruits a molecule called ZAP70 to the zeta chain, which actually binds to these phosphates. ZAP70 is important because it's also a kinase, and it phosphorylates protein phospholipase C, PLC. PLC is phospholipase C is associated with the membrane. It has regions that allow it to be associated with the membrane. And what it does is it converts inositol triphosphate, essentially the phospholipid, inositol triphosphate, inositol phospholipid, into two molecules. One of those is inositol triphosphate, and the other one is diacylglycerol, which it's basically cut. If you think about it, what it's done is it's cut the phospholipid in two, and this is the diacylglycerol component, and then IP3 is the inositol triphosphate is the other component. Both of these are signaling molecules, as we're going to see. And one, the diacylglycerol, because it's a fatty acid, has fatty acid groups and is a lipid, is going to stay in the membrane, whereas the inositol triphosphate is going to diffuse away from the membrane. When it does, the inositol triphosphate then activates the intracellular stores of calcium, and this is really important. A lot of signal transduction pathways are really one of the key modulators of that is calcium, and the release of calcium, either the sequestration of calcium or the release of calcium, is a big indicator. In this case, we get intracellular calcium released by the interaction of inositol triphosphate. This calcium is then binds to another protein called calmodulin, which specifically binds calcium very tightly, and essentially allows the calcium to be now useful as a signaling molecule. The calmodulin is now going to bind to a very important molecule. That molecule is calcineurin, and we're going to see this later because inhibition of calcineurin turns out to be important for development of immunosuppression. And so calcineurin will bind specifically to calmodulin, this calmodulin calcium complex, and they form this pretty interesting calcium complex. And what this does is it activates the calcineurin, and the calcineurin is going to act on a molecule on this transcription factor, nuclear factor that I mentioned before, NFAT. And NFAT is phosphorylated and found in the cytoplasm. Just generally it is. But when you have, with the action of calcineurin, what happens is calcineurin, this complex removes this from NFAT, producing the dephosphorylated NFAT transcription factor. And so when it does that, then the NFAT can actually migrate through the nuclear membrane. If it's phosphorylated, just to make sure you understand, NFAT can't cross the nuclear membrane. It has to be dephosphorylated. That dephosphorylation step is regulated by the calcineurin-calmodulin complex. Now, in the NFAT, once that transcription factor penetrates or goes across that nuclear membrane, it will bind to the DNA, and it also what is recruited there is another transcription factor, AP1. Now, these are two very important. Usually, when you find NFAT, you find AP1 bound to it. AP1 is fairly ubiquitous, has a lot of functions, as a lot of these transcription factors do, but AP1 is necessary for this and for the action of NFAT, and they form this complex on the DNA at the promoter regions of the cytokines, as we're going to see. Now that's one part of the mechanism. The next part is what happens up here with this DAG, diacylglycerol. The diacylglycerol is important because what it does is it recruits actually protein kinase C to the membrane, and there's an activation of that in which the protein kinase C is going to do an interesting reaction in which it's going to phosphorylate a molecule, and that molecule is the inhibitor of NF-kappa B. Now, what you find, the way that NF-kappa B is regulated is it's regulated by association with its inhibitor. So this molecule here is not active because it's inhibited. It has a complex, it's inhibited. But once the protein kinase C is acted on the inhibitor, what it does is that when it phosphorylates it, that is then released releases the NF-kappa B. So this can now go on and penetrate the membrane. So this is the regulation. So there's a phosphorylation of this inhibitor that then means that it will come off. And then what happens is it now can migrate into the cell, across the cell, the nuclear membrane. And so when it does this, it now can then associate itself with the promoter regions, and when it does that, what you see is then initiation and transcription of IL-2, and in some cases other cytokines. What I really want you to remember here is in the IL-2, in particular for IL-2, because that's what we've been talking about. So this is how IL-2 transcription is upregulated. And so again, just to recap, NF-κ is the transcription factor. This pathway diverges into two pathways in which you have inositol triphosphate, which leads to NF-κ dephosphorylation, NF-κ and AP1 binding to the promoter region. And then the other part of this molecule is the inhibitor is the activation of NF-kappa B, in which protein kinase C phosphorylates this inhibitor. The inhibitor then is able to cross the nuclear membrane associated with the promoter region, and thus turn on transcription of the complex. As we're going to see, there are a number of places in this pathway that are important. We'll see this later in terms of drugs that allow us to stop this process. And if we stop this process, we can stop an immune response and have a rationale for an immunosuppressive drug. So a couple of review questions. You might want to stop here and answer these review questions, and then the answers, of course, are on the next slide. So you might want to stop here, answer these review questions, and then go on to the answers. Okay, one of the last things we're going to talk about is, up until this point, we've talked about T-dependent AMI. In other words, the antibody-mediated response is entirely dependent on the interaction of T cells with B cells and antigen processing cells. Now, it came to be recognized that you can have some response, antibody-mediated response. It was discovered response without T cells. In other words, there were T-independent mechanisms. Now, some characteristics of these are you could see this response, and you could see an up increase of certain types of antibody in animals that are T deficient. Usually the antibodies that are produced are lower affinity directly without the need for T cell activation. They're not the highest affinity antibodies. One of the things that was noticed is most of the antigens for T-independent antigens have repeating subunits that are arrayed in regular sort of geometrical shapes, spacings. And many are high molecular weight polysaccharides like dextrans or LPS are ones that can do it. Other ones are virus capsids, particularly non-enveloped viruses, which have regular repeating units on their surface, and these can be strong inducers of a strong T-independent AMI response. And if an antigen processing takes place during this process, then the isotype switching will take place, and high affinity antibodies can be induced by the mechanisms that we've shown before. So it's turned out that this is maybe sort of a primordial way of initiating immune response. And it appears to be quite ancient, fairly ancient, as we know more and more about sort of simpler forms of life. It seems to be much more ancient mechanism. It may be one of the beginning ways in which the immune response was generated. So if we have, the way you describe it, so if you have a B cell and it interacts with, if you have an antigen in which you have a regular array of repeating subunits, 5 to 10 nanometers apart, as you can see here, they can interact with the B cell and result in a oligomerization. In other words, these antibodies get grouped or come together or cross-linked, or referred as oligomerization of these signals, and they create, because they all bind, right? Each one of these antibodies binding sites has the same, can bind the same antigenic determinant, and so when this binds, it can oligomerize the antibody, and this will lead to actually a bit of proliferation, some proliferation of these B cells, and some production of high levels of IgM secretion, and so you'll get some IgM that's secreted out of this. However, there's no memory that is garnered by this interaction, so you don't see any memory. So it's for a short while. You get some short-term proliferation of these particular B cells, and then you also can get high levels of IgM secretion. So almost sort of an initial protection that you can achieve by this mechanism. So just to take a look at this, if we then, for example, here's a virus, be a non-enveloped virus, interacts, you get oligomerization, and then what do you get is you get then IgM production from that cell, some limited production of this. Now, of course, the virus is a protein, so it can go into the cell and be antigen processed. And of course, then what you would get is a B cell activation and antibody production, as we have seen before. Everything you've seen before. So this we've all seen before, this B cell activation. And so there's sort of these two divergents. You get sort of this initial, you can get initial interactions with these molecules to give you production of some protective IgMs, IgM, to protect against the infection. And this can then be, over time, this will result, as the B cell gets further activated, into more sustained antibody activation and B cell activation. So if we were to look at the whole process, put this into our sort of schematic we used before, we can see if our antigen is now a virus, it would interact with the naive cell and give us IgM production. And then it could interact with the macrophage, of course. Macrophage would go through antigen processing. The same players are involved, Th2, Th1. The B cell can also go through antigen processing, producing Th2 cells, more Th2 cells, and memory cells. And then, of course, the activated B cell can then go on to do antigen isotype switching to produce now more IgG from the plasma, more plasmacytes, memory cells, and then further activation. So this is a way that you can get an initial response under certain conditions, under certain antigens like these antigens. Initial protective response while activating the longer-term antibody-mediated immune response, which we've seen before. So just a quick recap of this part. First, we've looked at the generation of antibodies to an antigen. We found that depends on initial and secondary exposure to the antigen. You now know why that's the case, because in the secondary response, you have those memory cells. The memory cells remember what has come into contact with both the B and the T cells. They're there. They can rapidly expand to produce more antibody and immune response to when there's a challenge from an antigen or a pathogen. And we also, sorry, something happened here. And we also saw that the IgM is produced first by Ig and then IgG, and we saw that class switching occurs, and now you understand where that occurs and why. We also looked at clonal selection and somatic mutation. This is production of more antibodies in the later stages, which have higher binding affinities. Clonal selection, again, is that initial stages where you're trying to really find those B cells, those antibodies that will bind to the antigen, as well as we go forward, find the T cells that have the T cell receptors that are necessary to bind to the MHC2 peptides. And then CD4 cells are really very important. As you can see now, they control B cell antibody selection and production. And so if we were to lose these cells, we wouldn't be able to really mount much of an antibody response, except through possibly a T-independent pathway, in which we just discussed, in which you can get some IgM produced that can be also protective. In the next section, part 4, what we're going to look at is how we can use antibodies and these antigens to induce protective immunity to microbial pathogens. And so that's going to involve looking at a few examples of how this whole process can be used for the development of active immunity, in other words, vaccinations, not passive immune response, but active immune response. So it's so critical to the development of vaccinations. Okay, so I think that's enough for now, and I will see you in class. Take care. Bye.